Welcome everyone to these invited lectures of the Josep Carreras Institute. Remember that those that are connected online, they can ask questions using the chat that we have there. We have today the great pleasure of having with us Professor María José Alonso. And it's, I think it's a great opportunity because yesterday she received the National Award in Research by the Spanish government. And that's really, it doesn't happen every year and every day. So congratulations, María José, for the award. She's a professor of pharmaceutical technology at the University of Santiago de Compostela in Galicia, in Spain, and her lab has worked a lot in the field of nanomedicine. And she has coordinated consortia by the World Health Organization, the Gates Foundation, and the European Commission. She's an author of more than 300 articles. Not only that, but she also worries about transfer of technology. And she has done more than 22 patents and started 3 startup companies. She's among the top 10 in pharmacology personalities in 2010. And she's in the list of the power list of the influential researchers in biopharmaceutics. She's the president of the Relief Society and editor-in-chief of Drug Delivery and Translational Research. Among other awards, she has received the Jaume Premier Award, the Awards in Excellence and Research by the Ministry of Science. She's a member of Royal Academy of Pharmacy, Royal Academy of Science of Galicia, and the Royal Academy of Medicine in Belgium, a national member of the US National Academy of Medicine. So today she's here to talk about her research. That's the main reason for her to be here. And she will explain nanotechnology as critical element in the development of mRNA COVID vaccines and maybe a few slides about cancer also. Thank you very much.
Good morning. I will have to stick here, right? So thank you, Manel, for the kind introduction. It is really a pleasure for me to be here today. I am really impressed by this institute in which you are working. I think you are very lucky to have the opportunity to work in such a great environment, scientific environment, and also nature environment. That's amazing. I was telling Manel I want to start my PhD here again. So anyway, when preparing my talk in the last few days, I was like, well, I'm going to the Josep Carreras Institute of Research. Why am I talking about COVID vaccines? I wasn't sure why I decided to give this talk, but it is certainly true that over the last 2 years, I have been kind of obsessed, as many people, about the pandemic, about doing something for this pandemic. And it is certainly true that I have been working for 3 decades now on the development of new forms of administration of vaccines. That was the subject of my PhD, my postdoc 30 years back at MIT. So I have been doing a lot of work on vaccines, but it is true that I am not an expert on vaccines. I am not an expert on immunology or virology. I am expert on nanotechnology. And basically, when we are trying to do is to use nanotechnology for solving problems of drugs or vaccines to try to make them better, more efficacious and less toxic. So we are a kind of solving problems lab. So this is my lab. We are based at the University of Santiago de Compostela, and actually, I think I'm going to try to be here. So the upper 2 floors are our labs, and this is an institute doing research on molecular medicine. We are also affiliated to another institute, which is just a couple of blocks near our institute. In the hospital. So I'm also affiliated to research institute in the clinical hospital, which is also very convenient for, having more, collaboration relationship with the medical people. And so this is my team, and obviously everything I'm going to say it was done by them, and I try to acknowledge properly all of them because this is their work. And as we always say, doing research is a collaborative activity, and we are all equally important. So during my presentation today, I'm focusing mainly on vaccines, although I would say a few words about cancer, and I will be able to, if you want, to extend on that subject. Basically, I will talk about serendipity, or how COVID vaccines were developed, then the history, a bit of the history of nanomedicine, and about the history of mRNA. Then I will share with you our laboratory experience on RNA delivery, and finally our contribution to the development of a COVID vaccine. So probably you have heard in the press or in the radio, you have heard the TV, that why this vaccine was developed in such a short time. And some people have said, oh, it was by serendipity, it was by chance that I found that these liposomes, things like that. So, well, serendipity is always there in science, this is true, but very often serendipity comes from the fact that there is a lot of accumulation of knowledge that eventually led to the apple coming from the tree, because of Newton, or when suddenly you have one idea. This doesn't happen as such, happens because there is a lot of knowledge, a lot of information that has been accumulated, and in a moment of high stress, like in critical situations like the global crisis, I mean, many people were just brainstorming and thinking, what can I do, and taking into account the information that has been generated. Eventually we led to a vaccine, or people led to a vaccine that is extremely successful, in my view. So everybody's talking about mRNA vaccines, and well, you know what mRNA, what can I say here about mRNA, but nobody talks about the carrier for mRNA, which are kind of liposomes. And in fact, they are called lipid nanoparticles, which are it's like a new version of liposomes. And in both cases, we can see that the origin of liposomes started in the 60s, and the origin of about the knowledge and certainly to envisage the potential of RNA started also in the 60s. So it has been the combination of both that has been made possible to have a very good vaccine, several good vaccines. So why do we need a nano carrier, a nano capsule, a liposome, whatever we want to call it, for mRNA delivery? Well, when we administer these nanoparticles containing RNA they can be administered by different modalities of administration, but let's say by injection, as we do with vaccines. What happens is that mRNA, which is very vulnerable to degradation, will be protected against degradation. In addition, we can achieve a targeted biodistribution. So eventually, in the case of vaccines, many of these particles will arrive to dendritic cells and produce the antigen and finally the protein. And we can achieve this intracellular control delivery, which is essential for mRNA. mRNA has to go into the cell. So this is another illustration that was presented. Nowadays, the ministry is asking us for dissemination of our projects. I think this is illustrated in the web of the Ministry of Science. And this is about how we make these nanoparticles, either for vaccines, or in the case of the project is for cancer, actually. But anyway, we mix mRNA with biomaterials, we make the particles, then we inject these, and then we achieve this intracellular delivery. There is an endocytic mechanism of uptake, then an endosomal escape, mRNA is released, and we produce the targeted protein. So this is how it works. So let me tell you a few words. I always think it is important to know where we come from, what was known, and when was the origin of the ideas. Well, the first people who talked about this idea of targeting, reaching the target, and about nanotechnology are these people here. So Paul Ehrlich, already at the beginning of the 20th century, talked about this magic bullet, like let's go to the target directly. So this idea of magic bullet has been there for a while. He was using this term in a different sense, trying to reach the microorganisms, and now we use it to reach the cells. But anyway, that was the idea, to be able to modulate the pathway of the drugs in order to reach the target in the most efficient way. And then Richard Feynman, who was also Nobel Prize in physics, was the first one who, talking about nanotechnology, he said holds great promise for the treatment of diseases, although he didn't have a clue about medicine. But he said there is a whole world of materials that we don't know yet, but probably they will have a certain application in medicine. So he was kind of a visionary. Both of them were visionaries. But then everything started in terms of nano carriers for drug delivery with liposomes. And actually it was a clinical doctor, a hematologist, Alec Bangham, who was studying the blood cells. He wanted to develop like artificial blood cells. And by serendipity, we can say, he found this kind of image in the microscope, and he was like, oh, what if I put a drug into these bilayers? How is this going to work? And then a few years later, other people more from the pharmaceutical technology field, like Peter Spacer in Switzerland, he was thinking about, rather than liposomes, let's use biomaterials, let's use biopolymers. And he made nanoparticles similar to liposomes. So as you can see here, everything started in Europe, actually. And this is just a summary of how the field has been moving. As I say, in the 60s, the idea of drug delivery started, and also the production of liposomes, and then polymer nanoparticles. Then it was the boom of biotechnology, and we realized how important biological barriers were for these biological drugs, which have great difficulties to reach their targets. Then 80s, 90s, it was the era of starting RNA delivery, in particular messenger RNA. And then nowadays, there are these two fancy terms, words, nanomedicine and precision medicine, that everybody's talking about. And obviously, all the evolution, I didn't put here the last discoveries, but all the evolution of imaging techniques were critical in order to understand, how these nanocarriers work. We started our lab in the 90s. So we have been, focusing on the formulation of biological drugs, mainly RNA, but also proteins, monoclonals. And actually nowadays we are following these different projects. We are working on theranostics, doing diagnostics and delivery at the same time, RNA vaccines, siRNA therapies, mainly of cancer. And we are starting now working on cell therapies as well. So this has been our evolution, just for you to have an idea. I went for my postdoc here. So basically, I have been living these years, seeing how this has evolved over the time. Well, I guess this can apply to many of us in my generation, because there has been such a big burst in research in the last decades. And nowadays, we are not talking about the future, we are talking about present and future. We have more than 50 nanomedicine formulations on the market. I have here a couple of papers with Bob Langer and other people from the European platform on nanomedicine, where you can see, the translational projection of this field, quite nicely explained. And nowadays, we have a huge variety, these are only some of them, of nano carriers for transporting biological drugs to their targets. So different kinds of nano capsules and liposomes and micelles and dendrimers and antibody drug conjugates, antibody polymer conjugates. All these structures are nanometric structures. fall into the category of nanomedicine. And this is just a list of nanomedicines on the market. And just to tell you that nanomedicines are being mainly applied in the field of cancer and infectious diseases, but there is no limit on the application, because as far as a drug has a problem to reach the target, here we are to help this drug. So for example, for ocular administration, and as I say, Abraxane was one of the first also going into the market, but also Pegintron for the treatment of hepatitis. I mean, there is no limit on the type of pathology that we can treat in a better way using nanotechnology. So the field has gone over the cycle, the hype cycle, which is very typical for any new. This happened also for gene therapy and RNA delivery. So at the beginning, there was a huge expectation, and then, you know, most big pharma companies started doing, you know, in their labs, nanotechnology, nanomedicine, and then they say, oh, this is not delivering, we are not putting anything on the market. So the interest went really down, and now we are going little by little, reaching this plateau where we can see that there are products going to the market. So at the beginning, also where we are now is going from academic exploratory research, although I do believe that it is still highly needed. We still know very little about what happens with the nanoparticles when they go into the cell, and we were discussing about that before. Understanding the mechanisms of interactions of nanomaterials with biology is critical. We don't know enough yet, but it is certainly true that we are assisting to a new generation of products with very interesting behavior. So, for example, Vyxeos, you know, this combination therapy, probably for the treatment of myeloid leukemia, and has a combination of cytarabine and daunorubicin. So they go together, and at the same time in the same place, and you can achieve the same delivery release profile or different release profile for both combination drugs. Another combination is with hafnium oxide nanoparticles combining with radiotherapy, also for the treatment of cancer, soft tissue sarcoma. And we have the first siRNA therapy that was marketed by Alnylam and Onpattro, which is also based on nanoparticles for the treatment of a rare disease. And finally, we have the COVID vaccines that nobody talks about the carrier, but the carrier, the lipid nanoparticles, have been essential. So curiously, the field of mRNA and the field of nanomedicine have evolved simultaneously. So at the beginning, the discovery of mRNA, the first liposomes in the 60s as well. Then the first liposomes containing mRNA didn't happen until 2 decades later. And then, well, mRNA synthesized in the lab. And finally, you know, the first mRNA vaccine tested in animal models. And finally, we are here with the 2 prototypes from BioNTech and Moderna. So talking about RNA delivery and nanomedicine, I do remember, I mean, I did my postdoc with Bob Langer, who, by the way, is going to receive in a few weeks the BBVA Award, Frontiers of Knowledge, with Carico and Weisman, and they will be recognized for creating technologies that together have led to the development of RNA vaccines. I'm very, very pleased about that, because what I said, nobody was recognizing the value of the delivery carrier. And Robert Langer is indeed a pioneer in the delivery of any kind of biological drug. Actually, I went to do my postdoc with him because I wanted to learn about biological drugs, and I was working on vaccines, but then I continued the collaboration with him, And I also did a lot of work with RNA, as you will see later. So he has been a key person, and he was beyond that. Robert Langer has not only a very high scientific profile, but also a very high business. He's a great entrepreneur. And he was the one who had the idea about Moderna. There were 3 promoters. He was one of them. I do remember when I was going there, I was between 91, where I did my postdoc, to 2000, I went every summer there for a few months. And I do remember seeing all big pharma opening labs for RNA delivery and then closing these labs because they say, oh, gene therapy is not working. The same happened with the nanomedicine field. And I was asking Robert Langer, what do you think about the future of this gene therapy? Now we don't talk about gene therapy, we say RNA delivery. The meaning of gene therapy has been used in different ways. And I say, well, all big pharmas are closing their labs. And he was like, don't worry, they will be back. And he said, here what we need is to have a joint venture of different big pharmas putting a huge amount of money into a lab, Moderna, and then something will come out. And this is what happened, basically. So he had this idea because it was a really breakthrough. The idea of using mRNA was just amazing. And he said, well, we need something big here. But I want to acknowledge also Peter Coolidge, who was the first who developed these lipid nanoparticles, especially adapted for RNA delivery. So basically, the technology that Moderna and also BioNTech is using comes from the same, originally from the same source, from the same person who is Peter Coolidge. He started, I don't know how many companies about RNA nanoparticles, making nanoparticles. Amazing, very good. We are having a meeting in a couple of weeks in his place in Vancouver to discuss about where are we going in the RNA field. So let me tell you what we have done in our lab very briefly. And I always show this slide, which is very simple. You have here different kinds of nanoparticles, and you see that they have a core surrounded by a kind of polymer or corona, polymer shell. And we say that we do a resonant design of these nanomedicines, taking into account first the nature of the drug to be loaded. So obviously the technology is going to be very different if we are working with a small molecule cytotoxic drug, or if we are working with a monoclonal antibody or an mRNA, very different. And then the nature of the barriers that we have to overcome. So if we are trying to have RNA delivery in dendritic cells or in other cells, if we are trying to reach cancer cells, if we are trying to administer by the ocular route, I mean, obviously the particles have to be different. So don't think about nanotechnology as a little box where I can introduce, a little capsule where I can introduce whatever I want. No, think about dressing your molecules. So each kind of molecule may need a different kind of dress, a different kind of suit in order to accomplish its function. And we try to use materials, biomaterials, which are recognized by their safety record. So biomaterials such as, polysaccharides, polypeptides, classical surfactants, oils, as simple as possible, because we want to be sure that our technology has a possibility for translation to the clinic. And this is very important. If we introduce a particle that doesn't have a safety record, it's going to be very, very difficult to reach clinical trials. So the different therapeutic areas in which we are working nowadays in Santiago is thermal drug delivery. We are also trying to build, to regenerate meniscus using our nanocarriers, delivering growth factors and delivering anti-angiogenic and angiogenic factors. We have been working, we are not doing anything now, but we have done a lot of work on RNA delivery to the eye. We are trying to treat autoimmune diseases We are trying to deliver proteins and monoclonals and RNA following oral administration. This is highly desirable for companies. We are trying to deliver RNA to the brain from the nose to brain delivery. And we have been doing for 3 decades work on vaccination, mainly nasal vaccination, and also working on targeting specific targets in the area of cancer. So just to concentrate on delivery of polynucleotides, because I mean, the vaccines are polynucleotides at the end of the day, just for you to know that we have been working initially on DNA, afterwards microRNA, siRNA, messenger RNA. So we do know how to dress, how to encapsulate these different materials and administer them by ocular, pulmonary administration, oral, nasal, intramuscular or intravenous. And the main areas of application is, I would say, cancer and vaccines. Brain is very difficult. We are trying to go there now with glioblastoma. We have a new project for theranostics of glioblastoma. Yeah, so these are the main activities nowadays. Half of the people in my lab is working on cancer, and a substantial group of people also on vaccines. So this is just the illustrations that my students make, different kinds of nano carriers that they design during their PhDs. All of them are different. And if you think about them, they are very similar to the virus structure. So this is coronavirus, this is an adenovirus. So there is also a core and a shell. We use proteins, we use polysaccharides, we use polypeptides, and this is the structure of the lipid nanoparticles, kind of liposomes used for for the vaccines. So in all cases, the size matters. The surface composition is extremely critical, and then the stability of these carriers in the biological media, in the bloodstream or other tissues, and then how they degrade over the time in the body. That's all very important. So focusing on vaccination, as I said, I started working on vaccination during my PhD postdoc 30 years ago, working with Bob Langer. We were trying to do single dose vaccination, and then needle-free vaccination following intranasal administration, and also thermostable vaccines. So one of our approaches has also been to design nano capsules with an affinity for the lymphatics, thinking about cancer and thinking about vaccines. We thought that, the classical way vaccines work is when you inject them and then the macrophages come to the place of injection and eat the vaccine. But we thought that there would be more elegant if we drive the particles to the lymphatics and we reach the dendritic cells within the lymphatics. So we have been doing quite a lot of work looking at how can we target nanoparticles to the lymphatics. And I think we have a video here. Let me see if I can show you how the particles accumulate. No. Well, basically what we were looking at is how we could modulate the nano capsules, what kind of polymer around the capsule and the size to reach the lymphatics and to accumulate in different regions of the medullary macrophages or other regions. And we could see in white, we have our nano capsules. We could see using this kind of microscopy, the accumulation of our nano carriers. So just for you to know that there is a way to modify the carriers to make them very attractive for lymphotargeting. Regarding the kind of vaccines we have been working until now, we started with hepatitis B, tetanus, diphtheria, very classical vaccines. We actually developed very good formulations that never reached the market because the idea was to implement them globally. That was the main purpose of the Gates Foundation. But eventually, these vaccines do work very well, in developed countries, and they do not work in developing countries because the way they use them, they share syringes or they don't administer the right doses. And although the encapsulation was working, I do remember this discussion with Bill Gates, who say, we made a mistake starting working with this kind of classical vaccines, thinking that their presentation in a nano format will make them much better, but even though they are better, nobody's using them. So we have to go for vaccines which do not exist yet. So then we started working with HIV vaccines in the form of peptides or in the form of mRNA. And lately, obviously, we have been working with COVID vaccine. So what has changed over 3 decades of work? So at the beginning, we were simply doing encapsulation of the vaccine in a PLGA matrix. And now we are achieving the specific localization either on the surface or inside the nanocarrier. And we are combining with immunomodulators. So the composition is a bit more complicated, but the response is much greater. So there is a significant advance in this field. So in terms of our efforts to polynucleotide delivery, well, we were the first actually reporting the encapsulation of DNA at this point in PLA-PEG nanoparticles, as you can see here, very small, and we have pretty good data with these nanoparticles. And then we moved to the use of polysaccharides, chitosan and hyaluronic acid for different applications, as you can see here, for example, in the area of cancer, in the area of ocular. So, well, just for you to know that we have a number of papers, this was also in the area of cancer, working with RNA delivery. But let's go finally to COVID. What did we do? So basically, just to remind you, we were collaborating with somebody producing the mRNA from DNA, then we encapsulated the RNA, then we did some tests in vitro showing how the nano capsules were accumulated inside the cell and were expressing the antigens, and then the T cell and IgG responses were evaluated. So 2 years ago, when we knew about the pandemic and we could organize a consortium, we were 6 people working full-time for more than 1 year, trying to develop all these different nanocarriers and comparing with the lipid nanoparticles that were being developed by Pfizer, BioNTech, and Moderna. So, you know, we can make many different kinds of structures. But just for you to know, in the lipid nanoparticles, what is essential? It's very simple. If you think about it, it's a very simple technology, but it was very difficult to find the right composition. So it has cholesterol. The yellow part is cholesterol, which is like a matrix, combined with other lipids, which are facilitators of the delivery of RNA inside the cell. We have a condensing lipid. This is the key of the patents. A condensing lipid that condenses RNA in a way, in a transit way. And then we have a stabilizing lipid, pegylated lipid around the particle to make them stable. So quite simple. However, thousands of compositions based on these ingredients have been prepared until now. So our consortium, it was mainly a Spanish consortium, although we had the collaboration also with a group in Brussels, Christian Tillemans. And the coordinator was Felipe Garcia from IDIVAX, and Montserrat Plana was also working with him. Then the University of Barcelona, Pompeu Fabra, Brussels, as I said, María Esteban from Centro Nacional de Biotecnología, and we at the University of Santiago de Compostela making the particles. So in Barcelona, using artificial intelligence, they designed the immunogen. Then in Brussels, they were producing mRNA. It was not easy to find anybody here in Spain producing mRNA, curiously. And then we formulated into nanoparticles, and we did, as usually, in vitro and in vivo validation. So, and then the next step should be, we didn't reach there, scaling up in clinical trials. So what we did in my lab was we prepared more than 300 prototypes with different kinds of structures and using different condensing agents for RNA, different surfactants, different oils, polymers, and lipids. And we were confronting these prototypes towards a target product profile. So when you want to have a good candidate, a good prototype, it's not a matter of cooking. Somebody think we have to make many prototypes and eventually some of them will work. So the target product profile is, we know that we want them very small, less than 100 nanometers. We know that we have to have sufficient amount of RNA molecules within the particle, of course, because in many papers you will see the number of active molecules into a capsule, into a particle is so small that you can only do in vitro studies. So the RNA has to be sufficient. We wanted to have it freeze-dryable because we knew about the problems with the lipid nanoparticles. And of course, we wanted the technique to be scalable and using materials with adequate regulatory requirements. So we produced more than 300 prototypes. We selected, taking into account this target product profile, 75 prototypes that we tested in vitro. And finally, we selected around 10 candidates. We are still testing for in vivo studies. And we tried with different RNAs at the beginning with model RNAs, and then with different kinds of mRNAs designed here in Barcelona. So the technique that we developed is extremely simple. And this is very important for the translation of the technique. I cannot go to the, let me see. No, I cannot make the video working. No, it's not important. Basically, what you see here is mixing two phases, and in a few minutes we have it. I mean, it's just the fact is an assembling of the different ingredients. So it's just a matter of finding the adequate recipe, the adequate composition. You have to look at there, yes. Yes, to click there. Yeah. So we have a phase, as you can see, and we add another phase, and we have the nano capsule. It's as simple as this. So here is, there are not chemical reactions. So it's all based on physicochemical properties on assembling of RNA with the different polymers and lipids. So you have to have the right proportion of each of them. And then we freeze dry them because we wanted them to be thermostable. And you can see here, this is the freeze dry product. And then when we add water, we have the original product. So this is the freeze dried nano capsules containing RNA, and we add buffer, and that's it. We have what we had at the beginning. And this is very important from the point of view of stability during storage. So obviously we have done lots of in vitro studies, as I showed you. This is to show that the nano capsules can go into the cell. You see here, well, this is actually not, I'm not sure if these are macrophages. You will know better than me. But this is the appearance, this is the structure of our nano capsules, and this is how they go into the cell. And then just to finish, to say that in collaboration with María and Carmen Gomez, we did, these are just some of the studies, but we have done at least 6 series of studies where we compare different formulations in terms of IgG, and we compare with Comirnaty, which is the formulation from Pfizer-BioNTech. And you see that we have similar or even higher IgG responses than the one obtained with Comirnaty. Although I have to say, I forgot to mention that for RNA to be stable, it's not only important to be encapsulated in liposomes, but there is also a chemical modification to be done in the mRNA sequence to make it more stable. We didn't use that modification, but still we had quite good data in terms of IgG and in terms of CD40 or CD8 cell responses. Well, just to make it simple for you, this is Comirnaty, sorry, it's not written, Comirnaty, and these are some of our formulations. In terms of CD8, Comirnaty did perform better than ours, but still we had some of them with good results. So you see, varying the composition of the particles with the same RNA, we have a total different response with the same RNA. So the nanoparticle really plays a very key role. And then when we, well, at the end of the day, you want to see what happens after a challenge with the virus. And we could see that the survivals, we tried homologous vaccination and heterologous vaccination with viral vectors. And in fact, we did see that with our formulations, 100% of animals survived, either homologous or heterologous. When we used only the viral vector, the response was not that good, so some of them died. So overall, these data are quite nice, but we have not been able to go into subsequent advances due to the lack of financial support. So we have quite good data in mice, and we hope to have the support to continue with either a COVID vaccine or another viral vaccine. I know that the majority of big pharma companies are now focusing on influenza vaccines and other kinds of vaccines, viral vaccines. So this is now a new field, and we will be there. So just a couple of words about cancer. Well, obviously, we identified the unmet need. Well, I don't have to say, you know, the problems associated to chemotherapy in terms of toxicity and limited efficacy, but maybe very important is the fact that chemotherapy only engages about 10% of all targets. And there are many targets, very potent targets, that have not been reached. And this is why there is this idea of undruggable targets. All monoclonal antibodies marketed so far, I think more than 40 just for cancer, either immunotherapy or targeted therapies, are all targeting extracellular targets. And people have not been thinking about how to reach the intracellular targets. These intracellular targets, you can reach them using a delivery technology. And we have been applying our delivery technology, which is illustrated here, you know, this kind of capsule. Obviously, the composition very different as compared to the vaccines, to achieve this intracellular targeting. And we believe is the first nanotechnology proven in vivo for the intracellular delivery of cool monoclonal antibodies. But today we don't have time for that. But the same similar kind of technology was applied for siRNA delivery for silencing specific intracellular targets. And we have done very little work. We selected this target, very challenging, which is the mutated Carreras, in particular this G12D and G12D mutations. There is a drug marketed recently for the G12C mutation, but these mutations have not been touched at this point. And so we have developed a monoclonal antibody that is able to reach these targets. I'm not showing you this data today. We started up a company to go ahead with these intracellular monoclonal antibodies. But we have also used, well, this is basically the principle of precision medicine in cancer. Obviously, we have to select the patients with the mutated Carreras, and we use an anti-Carreras antibody recognizing only the mutated Carreras, and we use this smart delivery. So Carreras is basically attached to the inner side of the membrane, and with our nano capsules, we go inside the cell, the monoclonal is delivered, and we reach the target. But since today, I thought, well, I should talk about RNA delivery, I can tell you that a similar approach has been done with siRNA, trying to silence this target. And this data, which were never presented, by the way, we could see that we could reduce the tumor growth, and this is illustrated here, when we used our nanoparticles with siRNA targeting Carreras, so silencing Carreras. And that's all, I believe. So what's next? We are working on, we want to continue. We hope to get the financial support to continue working in this consortium. We are also working with a consortium from Canada and United States on an HIV nasal vaccine, and this is much more advanced. We have already gone into the evaluation of the vaccine in macaques, and we do see protection against HIV. Let's see how this goes. We are trying to move into clinical trials now in collaboration with Manitoba, Wisconsin, and Harvard University, and we are putting now a lot of emphasis on personalized medicine using either monoclonal antibodies or RNA delivery. So this is where we are. And well, these guys are those who have done the work on vaccines. But obviously in cancer, we have around 10 people working on cancer now. And this is my full group. I thank them very much because, as you know, this is a collaborative work. And this is a collective intelligence that leads to great discoveries and great products. Thank you very much for your attention. And I would be happy to talk more about cancer wherever you want, because as I said, I'm expert on nanotechnology. The rest of the projects, all my projects are in association, in consortia, with other people who are expert in different areas. As you could see, for vaccines is for everything, always in the form of a consortium, because I have very limited knowledge about cell biology. Actually, this is what we're trying to go now, really collaborating with biologists to see what happens to our nanocarriers inside the cells. That is not that clear in many cases yet. Thank you very much for your attention. I don't know if it was too long.
Thank you very much. Was great and very different. So I like it very much. Some of the lectures we hear in this room. So I have extremely general and naive questions, in fact, because it's not my field really. But let's say I like it. Well, no, interesting for me the idea. This is physical, not chemical. When you use this sentence, no. So the cells that you use. The efficacy of the delivery of the nanoparticle depends on the cells. There is something in the cells, in different cells that make different the delivery, or not, or it doesn't matter because it's physical, you know.
You are totally right, and very few people are looking at that. So you have lots of people from especially with a chemistry background, looking at making very sophisticated nanoparticles, which are very sensitive to pH or enzymes and things like that. But then people have not looked at the different nature of the cells to see how this influences the degradation and the delivery. So this is going to be different depending on the cells, depending on the pH, but also the enzymes and everything in the cells, basically. Yeah. So this is going maybe different. It doesn't have to be very different, but it may be different. And this is something to investigate.
I also like very much the idea of that gene therapy now is called RNA delivery. So this rewarding a little bit. And related to this, so there are many diseases that there is a defect of expression of a gene. Mm-hmm. Okay, so then using these techniques, you can restore expression at a physiological level that looks like a normal wild-type cell. So you can do these approaches?
In theory, yes. In theory, yes. I mean, yeah, a number of groups around the globe. I mean, Moderna is doing quite a lot of work on on rare diseases, for example.
Yeah.
So I would say that everything is possible. However, I also I don't want to oversell this technology because, to be honest, with COVID, we were lucky. We were lucky. I mean, it's not that obvious that you will then produce the protein you want at the right amount and lasting the time you want. It's not that obvious. And it also depends, obviously, on the mRNA, not only the particle. And the different mRNAs are working very differently. So it may sound here like very easy, very beautiful, but it's not obvious.
And now there is a wall of the RNA modifications that for this delivery depends if the RNA is modified at this level, at this site or this site, maybe the efficacy is completely different. And you can play with that.
Yes. Yeah. So you have that part in which we're not expert, but there are very few good people here in Barcelona working on the chemical modification. Actually, most of the patents of Moderna are based on the chemical modification that they bought from the and then there is the part of the particles. Everything counts. But we were happy that we started from artificial intelligence with non-modified RNA, and we still had quite good responses. But yes, the modification of the RNA will add a plus, and I would say it's going to be necessary.
Thank you very much. Questions?
Thank you very much, José. Great talk. Just a comment to complement actually your presentation and a comment that Manel made a minute ago, is that I think that we can still get a lot of information from what we call nanomimetics. And we work in viruses, and we know how viruses use the cells. And viruses, at the end of the day, like a nanoparticle. And you know, I think that we can take a lot of advantage of the pathways that the virus use to infect cells. For instance, cell tropism, and understand how they internalize and see whether we can design nanotechnologies that might mimetize the virus pathways. So yes. I think there's a lot of way there, a lot of space there to do a lot of work as well. And the question I had has been, one of your slides has been about encapsulating antibodies, monoclonal antibodies. And this is something we are fighting to do in HIV as well and other viruses. And one of the limitations, especially for chronic infectious diseases, is the generation of anti-drug antibodies against the monoclonal antibodies. Mm-hmm. The question, I don't know whether you have any information on that, is by encapsulating antibodies into nanoparticles, would you preclude the emergence of anti-drug antibodies or not?
It's a very interesting question. Honestly, I don't know. I don't have an answer for that. It is quite interesting that there are very few people working on the encapsulation of antibodies. Everybody now is crazy about RNA, and so we found a niche there to become specialized in the encapsulation of antibodies. But we haven't tested that kind of. We think that we would reduce the immunogenic reactions, but we haven't gone that far. But if you have your antibodies encapsulated in your viral vector, we can compare to see how. But I definitely agree with you that we need to learn from viruses. We are trying to learn from viruses because the particles, but we try to make the particles as simple as possible. Because in the literature, you will see very sophisticated particles, even more than viruses, but they cannot make their way to the clinical trials because they are too difficult to produce and too difficult to control in terms of quality as well.
Thank you. Just, if you allow me, just one more question is, one of the issues with COVID has been that we thought with the first vaccines that we could avoid infection. And that it's clear that we can avoid a severe disease, but not infection. So in terms of nanoparticles, is there specific compositions or formulations that would allow mucosal immunity development rather than systemic immunity?
Another very important point, because we have been using, we started working on nasal vaccination, thinking that we will have mucosal immunity in addition to systemic humoral. And we do have both. And this is why we insist on the fact of having a nasal HIV vaccine. And also for COVID, I really wanted to try nasal, but my colleagues, they say we have to go for intramuscular because everybody is doing that. And unfortunately, we didn't have the financial support to continue with the nasal vaccine. But doing intranasal vaccination of macaques with HIV prototypes, we had very good IgA, IgG, and even, after 8 challenges, we had 80% of the macaques resistant. So I do believe that there is quite a lot of room for mucosal immunization using nanotechnology.
Hi, yeah, thanks a lot for this presentation. What do you think in the future, at which moment should we consider delivery systems in a drug development process? Right now, we always focus on finding a molecule or an antibody inhibiting our drug target. But can you say that then whatever we find, you will always find a delivery system? Or should we already consider this much earlier and maybe using worse drugs, but that can be delivered better?
Well, I think I was always complaining to my friends in big pharma companies that they are not putting emphasis on the formulation work. Formulation has always been development, and I say, well, there are many problems that we can solve, especially with nanotechnology. So I think, especially in big pharma companies, they are becoming very conscious about the fact that they need to think about the formulation, not at the end, once they have, the drug tested, fully tested at the preclinical level, because they are putting in the trash many molecules whose problems could be solved thanks to the nanotechnology, such as, stability, solubility, and also targeting. So it is true that sometimes they tell me, if we include nanotechnology, we are adding an additional hurdle. And I say, well, yes, you will only use nanotechnology if you need it. If the molecule is so easy that it goes where it has to go at a regular dose, no problem. The idea is that you go with nanotechnology only when you have a lot of toxicity, when you don't have enough efficacy, or when you really need it. As it is RNA, you really need it. You have to go with it. There is no way. I mean, you can go with a virus, you can go with a nanoparticle, but you need something there. You need a carrier, basically.
Oh, thank you. Fantastic overview. I just have 2 quick questions. Regarding RNA delivery, you mentioned you're optimizing the number of molecules per particle, but are you encountering any limit in terms of the length of the RNA that you want to deliver?
Well, not really. I mean, if the RNA is very big, we will have less molecules per particle. But yeah, I mean, there is no limit. We have even encapsulated viral particles, because as I said, we normally use the word of nanoencapsulation, and I don't know if this is the best word, because in my mind, it is more that we are dressing, we are covering the particles, we are packaging. My colleagues from Manitoba, they always say, let's package the antigens. So it's like kind of nano packaging with the adequate paper, with the adequate lace, something in that way. Mm-hmm.
And the other thing is, I was wondering whether, do your cargos reach the nucleus? Or does it depend on the type of particle?
That is very difficult. That is extremely difficult. A few people are trying, for example, with MIG, reaching MIG. That is extremely difficult because, as I said, now is a very good moment for collaborating with you, because we first had to generate the particles with biomaterials, etc. Then we needed to know about the biodistribution, the physiology, the anatomopathology. Now we have to go to the cell and understand what is going on inside the cell. And you will see thousands of papers of using nanoparticles and doing in vitro cell culture studies. Unfortunately, many of these nanopapers do offer very limited information because people will say, oh, nanoparticles go into the cell. For sure, I mean, in a cell culture, if you put the sufficient amount of nanoparticles, you will see them in the cell. You may have very nice pictures. But what happens there? How is the particle degraded? How is the release process into the cell? That is still quite unknown. I mean, we have hypotheses. We look at the mechanism of uptake. Okay, 4 degrees, 37 degrees, and we see if there is an endocytic pathway. We look at whether there is a receptor-mediated uptake, and then we look at the we see the particles inside the endosomal compartment. Then we look at the endosomal escape. And that's it. We don't know much more about it. So we need to know more, definitely.
Oh, hello, nice talk. It was about the endosomal escape, because they always, as you said in many articles, they do the cell culture and they say, we reach the endosomal escape, but sometimes it's really vague. Is there any formula that it really works for that, or in any study? Because
I mean, it's kind of brute what I'm going to say, but basically you create a lot of high osmotic pressure. You accumulate a lot of ions there. You create a lot of osmotic pressure, and the endosome explodes and releases. That's it. I mean, there are two ways. One is creating, accumulating a lot of ions within the endosome and making them to explode. And the other is to use surfactants that will permeabilize the endosome and facilitate the delivery of what is in there. I think, I mean, people talk about endocytic uptake. However, there may be also other mechanisms of internalization into the cell. The problem is that these mechanisms of internalization have been investigated in cell cultures, but how do you translate this to the in vivo situation is more complicated. So now we are trying to work with, as everyone, with you know, organoids and other tissue models.
Okay, in the interest of time, we should finish here. Not many questions. So again, thank you very much to the speaker.